This is question 2 of our core component mock, and it's mock number 1. So question 2, and it shows a fluid mosaic model of a membrane. And this is absolutely key to answering these questions. Fluid mosaic. And you should immediately recognise this structure of a membrane and some of the things. If you can't recognise this, then you haven't done your revision. Name the class of biologically important chemicals from which each of the parts X, Y and Z are made. So X, something that could sit on the membrane or within the membrane, so it could be extrinsic or intrinsic to the membrane. Those should ring a bell. Those are proteins. We don't need to give extrinsic or intrinsic or channel or carrier or whatever. It just says class of molecules or class of chemicals. So these are proteins. Y, this is our phospholipid bilayer. So but what class of biologically important chemicals is it? It's class is lipids, or we could say phospholipids there. So lipids or phospholipids. And Z, these bits that are attached to the protein. Well, if we're identifying actually what Z is, that would be carbohydrate. If we were saying, well, Z is this whole thing, then that would be a kind of a glyco for the carbohydrate bit, protein for the protein bit, and the full name will be glycoprotein. So carbohydrates, or you could possibly say polysaccharides as a smaller class of chemicals there. Or we could say glycoprotein. Make a label diagram to show the arrangement of molecules in part Y. So this is just part Y. We don't need any X's or Z's on there. So let's answer the question just in part Y. And so we're wanting the classic phospholipid bilayer. So the phosphate heads and fatty acid tails, and it says labelled, so you're not going to get all the marks unless you label it. So we've got phosphate head, and we've got fatty acid tails there. And so the marks were 1 mark for getting this double layer, this bilayer, 1 mark for getting the arrangement of these the right around, so heads out, tails pointing in, and the labels for the last mark there. Here's another classic experiment, so one that you don't actually need to memorise this experiment, but if you do enough exam questions, then you come across this a couple of times, widely used amongst the exam boards. So what have they done? They're giving you a bit more help in this stem of the question. It says model is described as fluid because the component molecules are free to move about, which will be useful later on. And molecules X are labelled with a dye. So molecules X, what are those? Those are our proteins. Okay, so we've got our bilayer Y there, so that will be Y, and X are some proteins. And this is the normal situation. And then this is a protein with a dye attached, so it's like one of these with a darker die attached like that. Okay, and why is that? Why have we done that? Just so that we can see it. It looks darker. And this is the start of the experiment. So all of the proteins have got the dark bit attached, and then they zap it with a laser. That's a fun experiment. And you can see that these are now bleached. So we've got an area that are bleached, and then they are now showing that it's not very dark anymore. It's kind of like a mixture, and you can see here got some light ones and some dark ones, and the answer is really in the question. So what's actually going on? So we need to say that our labelled proteins are able to move around in this layer because it is fluid, and it's really as simple as that. So our labelled molecules, and you don't have to say proteins because that was asking you what they were previously, but they are proteins. So X are proteins. These are able to mix with the others, are able to mix with the unlabeled molecules. No mark really for saying fluid because it already gives fluid there, but the idea that they can intermingle and move around. And then over the page, give three functions of a cell surface membrane. Quite a lot of people gave three things, but they were all the same point repeated three times in three different ways. He wants three sort of separate functions, so this is possibly a little bit more difficult. So yes, we say membranes control what goes in and out of the cell, but we need to sort of break it down a bit like that. What exactly can come in and come out and so on. So membrane, it's selective, so selectively permeable. That's what we really mean by it controls what goes in and out in the cell. So for example, we'll let in lipid soluble, e.g. let's through lipid soluble molecules. What other jobs do membranes do? Well, even from your GCSE you'll know things about phagocytosis and all. Let's do things that you should definitely know. Well, how about in our previous question we've got a membrane and we've got substance P being released out of the cell by exocytosis. So secreting chemicals. And I'll put, for example, exocytosis. And then, we could have a number of different options. We could have things like cell recognition. So in this glycocalyx, if I refer to another, like here, it's part of the question. If you're ever looking for suggestions, you can always flick back through what you've been given, and it might jog your memory a bit. Like you go, right, well, can I remember what that bit's for? So it forms a glycocalyx for recognition of cells. So I'm going to put that down. So cell recognition, and I'm just going to put glycocalyx. So we could also have things like receptor sites, but you might not know much about those yet. But those three things that you should definitely know about, and that's 11 marks. Again, people have generally got 7, 8, 9, 10, some 11s on that question. You should be getting good marks in that.